Welcome to the online classes of biology. So, dear children, we were studying about the prokaryotic cells, isn't it? So, we have seen that prokaryotic cells examples are bacteria, blue-green algae, mycoplasma, etc. Isn't it? PPLO, etc. And I told they are smaller than the eukaryotic cells, and the cell consisting of a plasma membrane. Then comes the cytoplasm. In the cytoplasm there is a region where you can see the genetic material is known as the nucleoid, isn't it? And then they are having plasma membranes in foldings known as the mesosomes. And then I also told you there are only one kind of cell organelle, that is ribosomes, and the ribosomes are different from the eukaryotic ribosomes, isn't it? Now let's see the cell envelopes and its modification in the prokaryotic cell. So in most of the prokaryotic cells, okay, there are particularly three layered envelope, okay, tightly bound three layered envelopes are there, which protects the bacterial cell, okay, or prokaryotic cell. So they include the outermost glycocalyx, okay, the outermost glycocalyx, then comes the cell wall, and then the plasma membrane. Okay, so these are the three layers of what? The envelopes, cell envelopes. Okay, now all these all these three layers are what is the function of them? They are helping as a single protective unit. Okay, their function is to protect the cell or the prokaryotic cell. And bacteria can be classified into two groups, like gram positive bacteria and gram negative bacteria. Depends upon their response to a staining process called as gram staining. Understood? So actually a scientist called Gram, okay, has developed a staining procedure, and in this procedure, okay, after the staining procedure done, if the bacteria retain the purple color, these are known as, that kind of bacteria are known as gram positive bacteria. And if they are not retaining the color of the stain, then they are known as gram negative. And why this difference comes? Because of the differences in the cell envelopes, okay, or there will be differences in the chemicals. Okay, so on that basis they respond differently. Okay, means one will retain the stains color and the other will not retain the stains color. Actually this color difference is due to the difference in the chemical composition of their cell envelope. And why it is significant? Because the presence of, or the difference in the cell envelopes, okay, is very much important as their mode of actions, okay, or their severity, okay, or their, what to say, their ability to make diseases and all will change. Understood? So only we are interested in this. Is it clear? So glycocalyx is the outermost layer, I said, and in that, okay, after that cell wall will be there, and then plasma membrane will be there. And glycocalyx, they differ in their composition and thickness, and sometimes it can be a loose sheath, okay, it can be present as a loose sheath known as slime layer, okay, slime layer. And in some other bacteria it can be very thick and tough, and then it forms the capsule. Understood? So the glycocalyx layer, if it is very loose, we call it as the slime layer, okay, it is not hard. Then sometimes it can be very tough, then it is known as capsule. Okay, then the cell wall, next layer is what? Cell wall, and that determines the shape of the cell, okay, and prevent the bacteria from bursting or collapsing. Then plasma membrane, we know it is the outermost boundary of the cell, isn't it? And it is same as that of the eukaryotic plasma membrane, okay. And I told some infoldings are there in the plasma membrane. They are known as what? Mesosomes. What are they known as? Mesosomes. Okay, so mesosomes are actually what? The extensions, okay, the infoldings of the plasma membrane, and they can be in the form of vesicles, tubules, or lamellae. Okay, different shapes it can have. So what are the functions of mesosomes? Let's see. They help in the cell wall formation, DNA replication, and distribution to their daughter cells. Okay, then respiration, secretion process, and it increases the surface area of plasma membrane. Okay, like this. So they help what various means to the bacteria or the prokaryotic cell to do different functions. Okay, and some other prokaryotes like cyanobacteria. Okay, you can see some other membranous extensions into the cytoplasm. They are known as chromatophores and they contain pigments. We have studied that cyanobacteria are what? The photosynthetic type, isn't it? So in them, in their cells, you can see some additional extensions and they are known as what? Chromatophores, and they contain the pigments. Is it clear? So these are what? The cell envelopes. Okay, the and it's what we have learned about glycocalyx. Okay, and then we have studied about cell wall and then plasma membrane. Okay, and you might have heard bacteria can be motile or non-motile. Motile means what? Which can move, and non-motile means which cannot move. So if motile, they are having some filaments. Okay, filamentous extensions from the cell, their cell wall, and they are known as what? Flagella. Okay, so flagella is what actually? They are the extensions, filamentous extensions from the cell wall. Okay, and depends upon the number of what flagella. Bacteria are again classified into different types. Okay, monotrichous, peritrichous, likewise different types are there. Understood? So when you see the flagellum, okay, flagellum is the singular and flagella is the plural. Okay, so flagellum, when you see, there are three parts. Okay, the filament, then hook, and the basal body. So these are the different parts of what? The bacterial flagellum. Understood? Bacterial flagellum consists of what? A filament, hook, and basal body. Filament is the longest part, okay, that extends out from the cell. Okay, then the hook, okay, and the basal body, they are present in the cell. Okay, now in addition to flagella, bacteria are having some what other extensions also, like pili and fimbriae. Okay, what are they? Pili and fimbriae. So pili's function is, okay, pilis are what? Long elongated tubular structure. Okay, and fimbriae are little more smaller, bristle like fibers. Understood? So this also you can see in some bacteria. Okay, and the functions of them is to get attached to the substratum. Okay, if it is found what found in the water bodies, to the rocks it can get attached, or to the host cells if they are parasites. Understood? And also they help to what means get attached during the sexual reproduction also. Okay, sex pili, they help in that. Their major function is to attach the bacteria. Okay, I have already said fimbriae are small bristle like structures and pili are what? Elongated tubular structures. Okay, so these are extra. Okay, some in some of the bacteria you can see, in addition to flagella, this factors also, and they are not taking part in motility, or they, for the movement, they are not having any role. Okay, now we will see what are ribosomes and inclusion bodies present in the prokaryotes. Okay, so in prokaryotes, ribosomes are seen associated with the plasma membrane, okay, of the cell. And you can see that it's made up of 15 nanometer to 20 nanometer in size. Okay, their size is that, and they are made up of two subunits, 50s and 30s subunit, and together they form 70s. Okay, 70s ribosome of the prokaryotic cells. Okay, and you can see what is the major function of ribosomes. Ribosomes help in the what are the protein synthesis. Okay, they help in the protein synthesis. So you can see many ribosomes are attached to a single mRNA, and this is known as polyribosomes or polysomes. So what are polysomes? Actually, in prokaryotes, you can see we will study this in next year in detail. That is how protein synthesis takes place. So during protein synthesis, DNA cannot means directly what give means it cannot direct the protein synthesis by its own. It needs mRNA, isn't it? So with the help of mRNA, when they form the proteins, you can see that RNA will get attached to the ribosomes first. Understood? And likewise, in prokaryotes, you can see many ribosomes are attached to a single mRNA, and that chain is known as polyribosomes or polysomes. Understood? So you can see this ribosomes are attached to where? To the RNA. Okay, many ribosomes will be attached to the RNA and they form the polysomes. Okay, so then next what is the inclusion bodies. Okay, inclusion, so the various food materials, okay, are stored. Okay, reserve materials or reserve food materials in prokaryotes are stored in cytoplasm in the form of certain inclusion bodies, and you cannot see any membrane that cover this, and they are seen freely floating in the cytoplasm. Okay, so examples of these are phosphate granules, cyanophysin granules, glycogen granules, etc. Okay, then gas vacuoles that are found in blue-green algae, purple and green photosynthetic bacteria. So these are what the major features of a prokaryotic cell. So from where we have seen prokaryotic cell, when you see what are the specialities of this? They'll have what from cell envelope. If you see, okay, the outermost layer is glycocalyx. It can be a slime layer if it is loose, or it can be tough, and then we call it as capsule. Then comes the cell wall, then comes the the plasma membrane. Plasma membrane is having some infoldings known as what? Mesosomes, that can be in the form of vesicles, tubules or lamella, isn't it? Then their functions are what? They help in cell wall formation, DNA replication, distribution to daughter cells, okay, respiration, secretion, to increase the surface area of plasma membrane. Understood? So likewise different functions mesosomes does, and the cells may have some the extensions, isn't it, like which help them to move or to get attached to the substratum. So the structure that helps in the movement is filament. Sorry, the flagella. Flagella is having what? Various parts like filament, hook and basal body. Then pili and fimbriae. Okay, they are what? Special structures. Okay, they are also extensions of the body. Understood? So about the outer part you understood now. Now next is what? Cytoplasm. Cytoplasm consists of what? Ribosomes. Okay, that is in ribosomes you can see that is associated with the plasma membrane. And what is the major function of ribosomes? Protein synthesis. So for during protein synthesis, one single mRNA will get attached to a number of ribosomes. Okay, now we call it as what? Polyribosomes. Understood? And then they includes what? Various inclusion bodies, okay, in the cytoplasm, which store the reserve food material, okay, like what? Phosphate granules, cyanophysin granules, glycogen granules, okay, etc. And in blue-green algae, you can see gas vacuoles and purple and green photosynthetic bacteria. In them, you can also see what? The gas vacuoles. Understood? And in cyanobacteria, I told one more extension is there, chromatophores, that carries the pigments. Okay, then there will be what? The DNA associated to a particular site. That site is known as nucleoid, isn't it? And what is the speciality of DNA? It will be single and circular, unpaired. Okay, unpaired chromosome in single one only, and it will be in circular shape. And in addition to that, some of the bacteria is having what? Another extra chromosomal DNA. Okay, that is known as what? Plasmid. Okay, so these are what? The smaller circular DNA found outside the genomic DNA. Genomic DNA is already there in the nucleoid. Is it clear? So this is the general structure of a prokaryotic cell. Okay, so there are no other cell organelles. No cell organelles are there except what? Except the ribosomes. We have learned that, isn't it? Ribosomes, what present is also different from the eukaryotic ones. That is, there are 70. Yes. Okay, so all these points are very clear, I hope. So we'll continue with the eukaryotic cell in the next class. Thank you very much.